compared to like trauma, I would walk to the mountain. Like I would walk alone and wouldn't even think about it.
Just water paper and there's another.
Okay, people. Okay, we're going to get started. Okay, folks, we're going to get started now. Thank you. Thank you. Great. So welcome everyone here in the room and also out in the wide world to our BME seminar series hosted by the Department of Biomedical Engineering at Columbia University. I'm Lisa Olson. I'm a professor in biomedical engineering and auditory biophysics in the otolaryngology department here at Columbia. So today and this year, we're offering this lecture in a hybrid format with both a virtual and a live audience. Our speaker, Deanna Thompson, is going to speak for about 40 to 45 minutes, and then we'll leave 15 minutes at the end for Q&A. So for our virtual audience throughout the presentation, please use the Q&A feature at the bottom of your screen, not the chat, to submit your questions, and we'll try to answer as many questions as possible during the Q&A session at the end. For our live audience, you can raise your hand the normal way. So today, we're excited to welcome Professor Deanna Thompson. Dr. Thompson is an associate professor of biomedical engineering and directs a research program in neural engineering with funding from NIH, NSF, DOD, the New York State Department of Health. She was a pre-doctoral NIH trainee in biotechnology with training in both engineering and the life sciences, earning her degrees in chemical engineering. Dr. Thompson was a postdoctoral research fellow at the Center for Engineering and Medicine with appointments at Harvard, Medical School, Massachusetts General Hospital, and Shriners Burns Hospital. She is a recipient of the J.D. Watson Young Investigator Award and was a recipient of the Rensselaer School of Engineering Research Excellence Award. Her lab is engaged in neural tissue engineering research for nerve regeneration, spinal cord injury repair, and neurodegenerative diseases. Welcome, Professor Thompson. So we can get started now. Turn it on.
Okay. Thanks so much. It is nice to be back in the real world and seeing faces as opposed to the corner of my bedroom wall. So I appreciate that. Oh, sorry. Thank you. Lots of wires, lots of wires. Okay, so I'm at RPI, and this is the Center for Biotechnology and Interdisciplinary Studies. And we're going to talk about some of the work that we've been doing. But before, I'm going to give a quick ad for RPI. Hopefully, of course, my battery just died, but that's all right. Oops. It's not advancing. Okay, so just to give a little shout out to RPI for those of you who are interested in graduate school, our application deadline is Jan 1st. We have a number of research thrusts at RPI that span. We have a relatively small department, but we have a big research department, core facilities and such. And so, it's a great place to live and learn in addition to NIH predoctoral training program. Not all of our graduate students spend every minute in the lab. There are lots of fun things to do upstate. Although you don't have quite as much city life as you have down here, but if you're interested in applying, please stop by our booth at BMES, or or contact me. Okay, so my lab is engaged in a lot of different research areas. We're interested, and I think we're going to talk today mostly about the electrical stimulation work, but ultimately I'm interested in asking questions of how different neural type cells, neurons, and neural stem cells interact with surrounding cells, support cells, right? Because we can study these singularly, but really how they interact plays an important role. So I'm sure many of you, right, you guys are all BMEs, are familiar with the anatomy of a peripheral nerve, but I like to show this cartoon because it's amazing, the structure, right? These individual axon nerve fibers are insulated by Schwann cells. These are the myelinating cell in the peripheral nervous system. These are bundled together to form fascicles along with a blood supply and connective tissue to form the nerve. So when there's an injury, right, this structure is often lost. If you crush an injury, right, these endoneurial tubes remain and there can be regrowth, and it has sort of a tunnel to grow through. But when you actually lose tissue, that becomes somewhat problematic. So for simple injuries or small gap injuries, you can actually suture together the nerve fascicles and allow for regrowth. But when that gap gets too big, you try to suture, you would form tension, you'd have scar tissue, and probably restrict the blood supply because you're pulling on that tissue, restricting the diameter of blood vessels. So what are the approaches that people have taken? For large gap injuries, the approach has yielded limited options for recovery, and basically fall into two categories. One are autografts, where you take a piece of healthy tissue from a healthy nerve and you actually implant that into that gap site. This provides scaffold, right, for those cells to migrate on. If you're doing an autograft, right, it contains Schwann cells that are there, and those Schwann cells are going to make good growth factors and support that regrowth. Alternatively, for patients who don't have access or there's a great deal of trauma to the area, you may be using guidance channels. And these guidance channels can be as simplistic as basically a hollow tube, or they can have superstructure. And there's a lot of work that's being done in decellularizing cadaveric tissue to maintain basically the structure that you would see in an autograft. But ultimately, there is, a lack of functional regeneration, and this is the area that a lot of people are interested in trying to solve. What are other issues that we need to overcome, right? Before I talk about that, let's talk about why these can actually spontaneously regenerate. So I mentioned before that the Schwann cells can produce great growth factors. And so this is just one myelinated axon, and you can see after an injury that these Schwann cells can actually, they actually phagocytose any inhibitory myelin, and they proliferate and they make growth factors. They can lay down a basement membrane. And this provides a nice scaffold for the regenerating axon to extend, right? And so this is why these small gap injuries spontaneously regrow. However, the other thing that we need to consider is some sort of time-dependent changes, right? And so there can actually be changes to the distal target if it takes too long. When you think about neuroregeneration, you think about it regenerating at roughly 1 millimeter a day. And so if you have if you have an injury distally, it needs to grow a very short distance. And when you're talking about something, a very high injury, it needs to grow much longer. And so if it's growing at 1 millimeter a day, everything distally is going to degenerate, right? The nerve fiber will degenerate, and those Schwann cells will be there releasing growth factor, maybe proliferating, removing that myelin. But over time, if they do not see an axon, you can actually have a reduction in the Schwann cell population. And you can actually, they become over time more poor responders to these growth factors. So we want to have things regenerate as quickly as possible, right? These are the challenges that in the space that we're working in. So to summarize, right? You can have slow axonal regrowth. We want to make them grow faster. And we can have a failure to repopulate the injury site with Schwann cells and even distally functional Schwann cells that are going to release growth factors, neurotrophins, as well as remyelinate the axon, because ultimately the axon gets there and it's never remyelinated, right? You're going to have poor function, and we would like that site to be revascularized. So as an engineer, right, we have a toolbox. And this is a pretty common cell and tissue engineering toolbox that one would look at. The literature is full of, different biomaterials related to neural tissue engineering, and obviously growth cones are responsive to soluble factors. Mechanics of the materials play a huge role. Mechanical interfaces between the two materials will play a huge role. Topography plays a huge role. But I'm going to really talk about this idea of support cells, and not to say that the others don't play a role. I think ultimately the ideal guidance channel is probably going to have multiple components, but we're going to really look at the how electrical stimulation and support cells influence neuronal growth. One of the things when I started doing some of this research, and really reading the older literature, there's endogenous stimuli that's present in the developing embryo, right? And this also plays an important role. So you can not only think about what we normally think about with electrically excitable cells like neurons and cardiomyocytes that respond and actually elicit a beat rate, but you can also actually have endogenous cues that affect all cells. And this could be translated over a short range by generating just short range depolarization, or you can actually have long range electric fields that form. So this can actually happen during development. And additionally, we can apply an exogenous stimuli. This could be AC or DC in a variety of fashion. So I'm going to first talk about the endogenous stimuli, because I think it's an interesting place for you to think about where all this came from. So this is a picture of a developing chick embryo. And we know that there are physiologically relevant electric fields that are important in these ion transporting epithelial, create these electric fields that are were thought to play a role. And during embryogenesis, they act instructively to guide the limb bud formation. So if you manipulate those electric fields that are present, you can actually have malformation of the limb buds. So during development, these are instructive cues. We know that in lower organisms, in work by Mike Levin and others, he's looked at morphogenesis in the planaria population. And it's actually, I like to show this because we did a stem cell program at Rensselaer, and these are one of the labs that we did. And I think that the teachers and the students really liked this lab. And this is a planaria. You can see its little eyes, they're light sensitive, so you can tell whether or not it's responding to light stimuli and whether or not those eyes are responsive. And using a special type of planaria following amputation, well, at all planaria following amputation, the neoblast will proliferate and migrate to form a blastoma. And, you know, how does a planaria know to regenerate? If we amputate its head, how do we know it's to regenerate the head with two working eyes? And how does it know to regenerate a tail, right? And Mike Levin has shown, you know, in his work, some really interesting experiments where he has basically shown that there's this polarity in the planaria, right? And so if you hyperpolarize the planaria following amputation, you can actually end up with a headless planaria. And if you depolarize the planaria, you can end up with a two-headed planaria. And what is so cool is that if you were to re-amputate both of those heads and tails, this is the ground state it's at. So you'll end up with a two-headed planaria and a one-headed planaria or a no-headed planaria. And you can actually split the axis to end up with a floor or eight-headed planaria. It's actually really cool. He has a whole website with lots of different experiments on there. And but I think this goes to show you that you know, if you dig down into the biology, there are short range cues and there are long range cues that instruct these cells, right, to do this. So when we're thinking about humans, or in the case of an experimental model, thinking about a rat, how do mammalian cells, can we, you know, how do they respond to either endogenous cues, or can we have them respond to exogenous cues? So many of these lower organisms are going to respond. I mean, they're much smaller, so the scale, right? The chick, when it's very small, right, the cues don't have to go as far, and as it's an adult, this becomes maybe diminish, right? Does it diminish with age? And so we wanted to investigate this in vitro a little bit, but to look at where the field was at, you know, there's been a lot of work where people have looked at chick, neuron, human neurons, and have shown no effect, some effects, varying effects to different voltage drops, right? People have also looked at, you know, animal models, as well as an animal model in the spinal cord. And this was early work that showed increased functionality by applying this electrical stimulation in the spine. And then they applied similar stimuli to humans, and basically it failed. It showed limited results. And one of the things as you're going from a rat to a dog to a human is obviously the size scale. What are those cells seeing locally? And are you giving them the same stimulus, right? Are you affecting the same volume of tissue that you need to affect? And so I think there is a need to understand how the cells are really responding to the stimuli and how all the cells present, not only the neurons, but how do the Schwann cells, the endothelial cells, and the immune cells, the macrophages, respond to the stimuli. Because we need to understand why in some cases things fail and some things succeed. Okay. This is work that was done by Tessa Gordon's lab. And this was a pretty cool study. She was looking at in vivo stimulation following a crush injury. So basically you had a carpal tunnel crush. They excised it to remove the crush, and they applied, some patients received electrical stimulation and some patients received no stimulation. And you can see that they showed that the number of motor units increased for the ones that received electrical stimulation. Now, remember, a crush injury, the structure of the nerve is present. You're not losing that gap. So it's a little bit easier, right? So we're actually interested in a more difficult place to return. But this is giving us really some confidence that this approach may have some merit. Okay, so we know that it can promote regrowth. We know that we want to see what's going to happen to these partner cells that are present. And we want to basically make sure that this isn't going to cause greater scarring. We're not going to have unleash a lot of very highly inflammatory macrophages at the site. Those are things that we were really concerned about. So let's talk first about the Schwann cells and the neurons, right? So we stimulate these in vitro. We initially started doing DC stimulation, and this is our first device that we actually created. And basically we had our control in our stimulation chamber. We could multiplex this, but this is just showing two possible chambers. We read out our voltage drop. We have electrodes that are placed at either end of the chamber, and we can monitor the voltage drop and temperature in the chamber over a period of time using LabVIEW. And so this is kind of, this was some of the earlier chambers that we developed. Our first experiment, we were asking, how do Schwann cells and neurons respond, and what's sort of the magnitude, right? If I'm adding, it's kind of like adding nerve growth factor, right? If I had 10 milligram, 10 micrograms, 20 or 40, how do the neurons change to that? And so similarly, what's more important, Schwann cells or electrical stimulation? How do the relative importance compare? And that's one of the things we wanted to know. So we cultured neurons alone on laminin coated cover slips, and we had Schwann cells where we seeded neurons on top, and we exposed them with electrical stimulation or without. We looked initially at a range of electrical stimulation from 0 to 200 millivolts per millimeter to see what would be supportive of Schwann cells, that we wouldn't kill the Schwann cells. And we basically ended up on this 50 millivolts per millimeter range. And I can talk more about that later. So using Neuralucida, we could actually quantify the outgrowth. And looking over at this graph, hopefully you can see that if we have Schwann cells, if we have neurons only, right, they grow a little bit. And if we have neurons growing on Schwann cells, they obviously close to double, right? This increase. If we electric stimulate the neurons without Schwann cells, it's almost the same amount as growing them with Schwann cells. I was surprised about that. I would figure the biological cell would probably do better than some sort of electrical stimulation, is kind of what my thought would be. But that was not the case, and we were pretty excited to see that. And when we put both cues together, the Schwann cells and the electrical stimulation, we see a further enhancement. So almost an additive effect. So we were really excited. And this is work that was done originally by Abby Coppice, who's now a faculty member at Northeastern University. And so the question that Abby asks is, how is the combination of these two cues manifesting themselves? And can we decouple that? So we decided, okay, we're gonna electrically stimulate the Schwann cells and then put the neurons on, right? We're not gonna stimulate the neurons. We want to just see what's going on. And so we ran an experiment that actually we were lucky that we were a little lazy. And so we essentially had Schwann cells, right? And we had Schwann cells that we didn't expose to electrical stimulation and did. And then we decided, okay, well, because we're stimulating them so long, by the time we set things up and whatnot, I was just like, we'll just put them in the incubator and we'll put neurons on them tomorrow, right? And so that's basically, that's what we basically did. We put the neurons on and we let the neurons grow overnight. And then the next day we fixed the cells, right? And so we could fix an image, we could quantify to see whether or not the Schwann cells changed in orientation, and we could actually see the neurons and quantify the outgrowth. And so I'm going to show you the data in a little different way, and this time a polar histogram, because we saw initially some biased outgrowth. So we wanted to see if we could see this biased outgrowth. But this is the original neurons alone, right? They grow, right? They're happy, they're fine. If we put them on Schwann cells, they grow a little bit more. Or if we electrically stimulate, they grow the same amount. And we can put them on these on the Schwann cells and neurons together that we stimulate at the same time, and they grow a little bit more. So I was surprised to see that when we electrically stimulated the Schwann cells and then put the neurons on, right? The neurons have not been electrically stimulated. We see this crazy amount of outgrowth, right? And we kind of asked ourselves, you know, what was going on there? Well, it turns out that when you electrically stimulate the Schwann cells, you are actually having them release nerve growth factor. They start producing and releasing nerve growth factor. So in our area of fortuity, after the stimulation, we would actually see the neurons basically right around here, and we'd let them grow for 12 hours. So we'd be in this kind of sweet spot. We ended up lucking out. And over a period of time, this NGF goes back to basal level, right? And we know that this change in NGF is not due to the number of cells, because we normalize for that, and they follow a very similar trend, maybe a little bit more, but not statistically different from that. So we've looked at the mRNA of BDNF, GDNF, and other neurotrophins. The Schwann cells that we're using are isolated from a mixed nerve, and the way that they're cultured, they basically are not, you could actually get sensory specific and motor specific. Ours are just kind of a mixed population. And so we would not expect them to make a lot of BDNF. If you isolate a pure population and don't culture them with a lot of FBS, you can actually get them to make other neurotrophins, and ours typically don't. So you can see, if you're looking closely, here I've now said AC stimulation. So we move from DC stimulation to AC stimulation. We're applying 20 hertz, the same sort of amplitude. We looked at whether we stimulated them for 8 hours, 4 hours, or 2 hours. They needed to be stimulated a little bit more than 2 hours, but 4 hours seemed to be the sweet spot. And so 8 hours showed no improvement. So that's kind of how we kind of are narrowing in there. Well, one of the things that my postdoc and myself were thinking about is, could we actually think about how we think about control release, right? Instead of putting spheres in there that are releasing NGF, could we actually stimulate those local cells and actually cause them to release nerve growth factor? And that's basically what we did. We could run an experiment where we could actually stimulate multiple times, and we could see reproducible increases in NGF. So instead of thinking about, I'm stimulating because the neurons right? Those axons are growing faster, which is a good thing. But I'm also stimulating and I'm affecting the Schwann cells that are there, and they're releasing more neurotrophic factor, and that could possibly be used as a benefit as well. And so this is kind of another way to think about this. We wanted to see whether or not it was due to NGF or some other factor. And so we did a simple experiment where we were comparing electrically stimulated Schwann cells, where we applied an IgG versus an anti-NGF. And basically we could see a reduction in outgrowth. So it in all likely is due to the NGF, in addition to probably some other factors that we would have to do a larger screen in order to basically see that. How are the cells responding to this, right? How do the Schwann cells get the signal and then respond to this increase in NGF? Well, we looked at actually at these voltage gated calcium channels, and by blocking different calcium channels with small molecules, we found that the T-type voltage calcium channels seem to basically stop this increase in NGF. So it's probably mediated in that sort of way. And a lot of when you think about endogenously stimulating cells, people can also use small molecules to basically trigger these voltage gated ion channels instead. So instead of doing electric stimulation, you could actually apply a small molecule that would cause potentially a similar increase or change the voltage gated calcium channel in such a way that you might elicit the same sort of thing. And we haven't actually explored that as much, but I know other people are. Okay, this is all well and good, right? It's in a petri dish. Who cares? We want to apply this sort of thing in a three-dimensional space. And so we wanted to do this in sort of this is just a summary of what kind of we did, but we'd like to do this in a 3D biomaterial that maybe more mimics what you would actually apply in an animal model. And we want to see whether or not these cells will migrate, and whether or not their migration can actually be improved in a three-dimensional hydrogel. And so what we ended up doing, we did a pretty simplistic experiment. We basically created, we did this with endothelial cells. We also did this with Schwann cells. And we created a concentrated region of cells. And we then surrounded it with an acellular matrix, right? We could then apply electrical stimulation, and we wanted to see whether there was a difference between the stimulated cells and the non-stimulated cells. And basically we kind of run the same sort of experiments. And so we can see that following electric stimulation, we actually see sort of these cords of Schwann cells migrating out. And these were they migrated out in all directions. So we didn't see a directional response, even though we were applying DC stimulation. Some people have said that they've seen directional response. We didn't see that as well. So we could also apply this with AC stimulation and not necessarily worry. But this is kind of very common to what you would see in migrating Schwann cells. They kind of migrate out in these little cords in these times and produceably, lay down basement membrane for cells to migrate further. And so here we basically wanted to show, we quantify the electrical stimulation, and we see greater migration of Schwann cells and greater migration of endothelial cells actually in response to electrical stimulation than not. Again, no directional manner. So they're migrating faster. And the question is, why are they migrating faster? If we looked at MMPs, right, because this is a collagen-based matrix, and we see that the electrical stimulation basically turns on some of these MMPs, and it turns down some TIMP inhibitors. And so these cells, these Schwann cells, are able to actually break down the matrix faster, which allows them to migrate faster. And similarly for, if we look at this 8 hours and then 24 hours later, we see, you know, different MMPs turning on and the TIMP inhibitors still going down. So this could be a mechanism as to why that these cells are migrating through. So if you want cells to actually migrate through a material quicker, part of the reason is these Schwann cells are changing and they're actually being able to degrade their matrix much faster. We saw that we could actually look at this. We applied the calcium channel blocker, and we basically see that it shuts down NGF, but the cells still migrate. So these things are being mediated through completely different mechanisms, and we haven't really explored that as well, that much further. We asked a similar question with endothelial cells, and endothelial cells are also responsive to MMPs. We see this increase in MMPs, maybe possibly allowing them to break down their matrix much quicker, slightly different MMPs. And so, but not this reduction in TIMP inhibitors. So that is that. When we apply electric stimulation with or without Schwann cells, we see a benefit. So here is where we can't do that 2D experiment. We can't electrically stimulate the Schwann cells with the neurons not being there. So we can't pre-stimulate them. The only thing we could potentially do, and we haven't yet done it, is we could stimulate the Schwann cells and then see them in the gel. But you would probably have to wait too long before, or put the neurons on top, and then they probably wouldn't actually go into the gel. But in any case, we can see that the neurons only grow only so much, extends only so much. Ones with stimulation or Schwann cells increase much further, and co-presentation of that results in a further increase, but not additive as we saw in 2D. So there is more work to do there. But it's opening our eyes a little bit to how we think about things, right? So if you're only looking at your end result as the neuron, how far the neuron's grown, but you're not looking at what's happened to these other cells, you might be missing opportunities to improve that function. So exogenous electrical stimulation, right, can increase neurite outgrowth. We can increase both the rate of migration in a biomaterial, as well as the release of NGF, right, to further support outgrowth. And we see these changes that are observed in non-glia that can actually increase the rate of migration of endothelial cells. And this could actually maybe play a role in terms of revascularizing a tissue, right? By electrically stimulating, can you actually get endothelial cells to invade that tissue in a faster rate? So one of the questions we've always been asked is the immune response altered, right? We're applying this electrical stimulation. Are we making these macrophages very angry, right? Which is going to undo all the things that we're currently doing. And I had a master's student working on this for a very short time, and I'll kind of end the talk here. So I probably don't need to show you this, but you know, in general, people have thought about macrophages, classically activated macrophages, and alternatively activated macrophages as existing on a scale. In reality, it's way more complex like that. You really should think about it as kind of a color wheel, where they can exist in lots of different states. And these macrophages plays an important role, right? In terms of host defense, cytokine production, inflammation, wound healing, and matrix remodeling. If we have too many M1s, right, we have a lot of scarring and whatnot. But if we have too many M2s, we can also have problems. So we need this balance during the regeneration process. And I'm going to skip this. Well, other than to say that macrophages obviously respond to lots of different stimuli, cytokines, right, topography, matrix, whatnot. So it actually, it can respond to lots of different stimuli, similarly to neurons and Schwann cells. So we wanted to ask this because macrophages are kind of plastic. Can electrical stimulation switch their activation? right? Is it going to cause, if I'm applying electrical stimulation for 4 or 8 hours, am I going to make things angry, right? I'm going to make things very M1, or can I induce a more M2 response? And so we did some simple experiments with a cell line, right? As a way to just generate some preliminary data. And so we seeded these M0 cells and we could stimulate them with LPS, IL-4, or no cytokines added. And this would be kind of our control baseline. These would be induced for a more M2 phenotype, and I'm saying M2 phenotype with quotation marks because this is a really simplistic system. And then M1 phenotype. And then we can actually culture them for a period of time, and then we can electric stimulate them or not. And then we can collect their lysate and we can look at how they respond post-electrical stimulation for up to 48 hours. And we basically, we first wanted to see whether we can induce a phenotype at all, right? And so we looked at, in terms of INOS for, inducing this M1 phenotype, and we see with no cytokines added, we see nothing. IL-4, there's a small response, but really we see this big increase with LPS added. So we're okay. We're willing to kind of move along with that. Similarly, we're looking ARG1, and we can actually see this huge increase with IL-4. So we're using these two as markers of its transition from its M1ness to M2ness. And again, this is a very cursory look, just preliminary experiments going on by a master student, Catherine Kearns. So we first did this experiment with the M1s, and we basically add our LPS. We culture them for a period of time, and then we change the media, and we electrically stimulate them, and then we collect the lysate over a period of time. And we basically see that we basically are looking at this fold change, right? And we see INOS is always pretty high, regardless of whether or not you have electrical stimulation. So those the hash marks are the electrical stimulation, but we're not increasing we're not increasing its M1ness, right? But we do see this increase in sort of ARG1, right, with the electrically stimulated one. So we're asking ourselves, are we kind of pushing them a little bit toward this M2 phenotype? Maybe yes, maybe no. The answer's still out there. Similar experiment with M2 phenotype. We do the same sort of thing, and we actually we don't see actually any statistically different change in INOS. Remember, this is our M1 phenotype. Here, this is not statistically significant. We can never get these error bars very well. Catherine had a hard time with this 4-hour time point, but I show you this here. But we see this change in ARG1. We're pushing these cells more toward more M2, pushing them even further toward M2. Is the way kind of we thought about this, and we need to explore whether or not we're actually changing the cytokine profile coming out. So so at the end of the day, we looked at electrical stimulation. We explored how Schwann cells, how neurons, how an endothelial cell line and a macrophage cell line all responded to the stimuli. And we can see that these cells do have some response, which is exciting. And this is another tool in our toolbox that then we can actually potentially apply. So I feel like electrical stimulation has a profound impact on these cell populations that shouldn't be ignored. People are applying electrical stimulation for brain stimulation, for to treat lots of different disorders, and understanding how that stimulation impacts not only the neural tissue, but the cells within the neural tissue, I think is really important to fine tune that. I think electrical stimulation could be used in a time-dependent manner to promote regeneration, depending on your ability to deliver that stimulation. And in conjunction, it's very easy to do this in conjunction with biomaterials or even other other tools in our engineering toolbox. And so further research is needed to explore this, and obviously translation to a injury model is kind of the next step. So with that, I think I wanted, hopefully didn't go over. Maybe I did. I don't have my clock here. I'd like to thank the students who did the work. A lot of this work was started by Abby Kappas, Ling-Sha Zhang, is currently at Regeneration, and Catherine Kearns, who left with her master's. Collaborators that I talked with a lot at RPI, people in ECSE who helped get us going. And obviously my other collaborators in the department, Ryan Gilbert, Sally Temple in the area, and Vivian Mouchouar at University of Alberta. So with that, I'm happy to answer any questions.
You know, I think at this point, we're going to want you to stop sharing.
Okay, perfect.
So, before taking questions, I just have a couple of quick announcements. Let me. Okay, so but anyway, thank you, Professor Thompson, for the great presentation, and in a minute we'll get to the Q&A. But I just want to draw everyone's attention to our next speaker, Professor Trina Arinza, who will be here on September 23rd. And remember to follow us on social media to receive the latest updates. You can see our social media information on the slide, and you can always access the registration information for our BME seminars. So I think we can start with any questions that we have in the audience, and then we do have a couple online, but you guys can go first. Any it was a fantastic presentation, by the way. I have some questions. So, yeah.
Okay, this may be very, very rudimentary question, but you talked about the research you were doing where you had the Schwann cells, you essentially stimulated the gel and then and you said that it was tougher for the neurons to grow in that environment or in that experiment.
So I think I misspoke. So I would basically, because we couldn't run the same experiment that we did in 2D, right? You couldn't electrically stimulate the Schwann cells without the neurons because everyone's there, right? Everyone's at the party. So what we did is we put Schwann cells in the scaffold, and then we put a ganglia in the center of the scaffold, so then you could see really nice outgrowth. And I sorry, I didn't give that experimental detail.
Thank you. Great talk. I just wanted to ask you, did you also study the effects of stimulation on other myelinating cells, like oligodendrocytes or something like that?
I haven't. No, we have not done that.
Maybe I'll go to one of our questions. I don't know if I really need this. So and actually you can also read along.
Okay,
if you want.
sure.
So we can start with the first one that came in early. From Praveen Tarkali. Thanks for enlightening presentation. How does the genetic engineering of neural stem cells affect the rate of neuronal regeneration?
So, so we haven't actually, we haven't done a lot with neural stem cells. I mean, we've done some work with endothelial cells and neural stem cells. We are working with human iPSC neural stem, Schwann cell. We do a disease Schwann cell that are iPSC derived, and then we electrically stimulate those cells. So, but we haven't actually done neural stem cells where we've electrically stimulated.
So another chance for the audience before I go to, oh, okay, yes.
Where do you get the cells?
We isolate them.
From what?
So we isolate them from rat neurons. So we can actually isolate the Schwann cells, and we purify them, and we can freeze them down and use them at will. And we can actually do the same thing with the neurons. To reduce the number of animals, we isolate and dissociate the DRG. And we can actually, if we're not going to use them for a long time, we can cryopreserve. You lose viability, but they actually, the ones that break out go great. There's also this hibernate solution that you can actually just put whole DRGs and stick them in the fridge for up to like a couple days, and you can run experiments. Brain Bits, I think, sells it, and it works awesome.
So I'm gonna go to the question, that's my question. Just by Olawale Alimi. Thank you for the insightful talk. I'm wondering what guided your choice of 50 millivolts per millimeter. Also, could there be an advantage for increasing the electrical stimulation above 50 millivolts per millimeter? Does the current also matter? Thank you.
Yes. So good question. So initially when we did this, so the question was why 50 millivolts per millimeter? And does also the current matter? And could there be advantage for increasing this above? So when we were thinking about doing this, if you're applying electrical stimulation to a human patient, we're thinking about, you know, if you're below a certain threshold, there's people will say that, like, you know, in terms of like transcranial and other types of stimulation, things are like are willing to, a patient's willing to kind of, you know, succumb to something like that. But once it goes above a certain level, so we wanted to stay within what had been has been measured and reported physiologically in the cell during development. We didn't want to go above that, right? So you'll see papers where people go really, really high. And we kind of wanted to stay at that lower range, because if you actually apply DC stimulation in cell culture, you can see the pH change that occurs very quickly. So you're not going to be able to stimulate for a very long time. So that was practically one of the first reasons. What we ended up doing is we looked at from 0 to 200, and we looked to see where the Schwann cells would live, and look, you know, there's no basically apoptosis or no, everyone looks healthy and they proliferate. And so we basically, I think up to like 120 or 150, everyone looks pretty good. And then we looked at a range of neurons, and we found like the neurons within a certain range would grow more. And we just kind of like basically ended up there, but we didn't do a lot of, our throughput wasn't high enough. And the tracing was cumbersome enough at the time, you know, that there could be a better spot. It could be a better field.
Do you know that 10 millivolts per millimeter isn't high enough?
Yes.
Okay.
Yes.
Yeah.
So I think like 10 we tried. I don't know that we went to 25, but like, yeah, I think 20 millivolts per millimeter, I think wasn't high enough, at least for the neurons that we were using.
Yeah, it seems like a low number to me. 50
Yes.
millivolts.
Yeah.
So, okay, interesting. Yes.
So in your opinion, which method is viable for like human translation long-term?
Did you hear that?
No, I didn't hear
Speak
that.
up a little bit.
I said, in the long term, which one would you be the most viable? Your opinion, like electrical stimulation or optical?
I think both, potentially. I mean, both.
Catch that, or can you repeat the question?
Which would be more better, either basically electrical stimulation or optogenetics. I mean, I think depending on what you're doing, potentially both. People are also using LIPUS, like low intensity pulse ultrasound, and actually seeing that Schwann cells and neurons will actually grow because of that type of stimulation. So depending on what nerve you're trying to hit and where it is, right? You know, ultrasound potentially can go to a different place. You have a nerve couple electrode that maybe if you can actually stimulate, do you want to just stimulate the injury site, or do you want to stimulate distally? So if you're doing ultrasound, you could potentially stimulate the range, right? And it kind of depends on, I guess, what you're trying to get, right? If you did like ultrasound or some way to actually stimulate over the range of that nerve, because I would imagine that, you know, by stimulating, and this is something we don't know, but, you know, if you're talking about peripheral nerve interfaces, not even regeneration, but interfaces, you know, how do you make that interface very healthy to interface with the prosthetic? And is there some way that you can kind of provide some stimulation to kind of make a really good connection.
Kind of to that end, like have these or like can these methods like be applied for like human like translation or human methods, or like has any work been done?
So people have done this with the electrical stimulation, carpal tunnel, where it's just a crush injury. So people have already applied that. People have done clinical trials where they've stimulated the nerve before they've sutured it. And actually that's some of the work that Tessa Gordon has done. And she actually showed that in a mixed nerve, it actually increased the rate of motor ending up in motor and sensory ending up sensory, which is kind of cool. So yeah, people have done that. I think you might need more stimulation over a period of time in order to get the most bang for your buck, because usually there's a greater rate of regeneration and then it kind of plateaus. So.
Then what are the lasting effects of this type of thing?
What do you mean?
So as far as growth goes, so yeah, you do stimulation, you have this, you have growth. Does the neuron stay the same size throughout? Like, do you have to re-stimulate when
More growth?
Yeah.
I would imagine there's going to be a period of time that, so we're looking at neurons practically for a very short period of time. And the reason why is because if we want to trace them, right? But that's a good question. I mean, presumably if we're stimulating the Schwann cells and they're making more NGF, right? More than the basal level, we should see more outgrowth. So in theory, we could stimulate that same population over and over again, and we should see greater outgrowth over a period of time. So hypothetically, but we didn't look at that.
I think this will be our last question, so.
So you mentioned that this information is used Schwann cells, so I was wondering what was like what was the data sharp in that? And what were the differences that were identified in contrast to the healthy Schwann cells that you previously helped?
Right. So this is a new project. And we're interested in neurofibromatosis type 2. And so this is unlike type 1, you get these bilateral tumors, and patients typically come in with hearing loss. And then there's usually a second tumor that's found, potentially. And so kids can actually lose hearing bilaterally because of these large tumors. So we're interested in actually what are the differences between the normal Schwann cells, the control cells, and then the NF2, and then what makes, how do they get that second mutation such that they, turn into cancer. So we're interested in that. And then how the tonic activity of the nerve may influence that growth.
Okay, well, let's thank Dr. Thompson.